Good afternoon, everyone. I hope you all had a wonderful experience during the first session of our faculty development program. Now, moving towards the second session of day one, let's welcome our expert for this session, Professor Sanjay Kumar Singh, sir. He is currently working as a professor in the Department of Computer Science and Engineering at IIT BHU. Sir holds a PhD in Computer Science and Engineering along with BTech and MTech degree and professional certification from Novel Netwear USA. His key research areas include biometrics, computer vision, pattern recognition, and artificial intelligence. With over 100 publications, editorial roles in reputed journals, active participation in international conferences, he is also a member of prestigious organizations like IEEE, ACM, and CSI. We are deeply honored to have him with us and look forward for gaining valuable insights through his session. Now, I would like to call Professor Sanjay Kumar Singh sir for this session.
सर आप म्यूट हैं अभी.
Sorry. Yeah. Okay. So, I'm audible now?
Yes, sir, you are audible, sir.
Okay. So I'll just share my screen.
Sir, you have rights to share your screen. Yeah, it's visible, sir.
It's visible?
Yeah, yes, sir, it's visible, sir.
But yes, some issues are there. It's blurred.
Abhishek, coordinate for the same.
Just please, some issues are there still.
सर स्क्रीन शेयर प्रेजेंट नाउ पे आएगा सर इसको फुल स्क्रीन F10
अच्छा फुल स्क्रीन just a minute.
F5
just a minute. Can I make it this entire screen then? The entire screen is not.
सर F5 प्रेस कीजिए स्क्रीन शेयर पे जाके F5.
Is it visible? No, I don't think so. It is visible.
No, sir. Yes, sir. Now press F5.
Just a minute. No.
सर इंटायर स्क्रीन करके भी आप उसको शेयर कर सकते हैं सर.
I don't know what is the issue, but is my screen visible to you or not?
Yes, sir. Screen is visible. Sir, full screen नहीं है.
Full screen
नहीं है तो आप F5.
But I'm not able to do it full screen.
Yes, sir.
Some issues. I don't know why I'm not able to do it.
सर एक बार क्लोज करके वापस से सर फुल स्क्रीन इंटायर स्क्रीन पे शेयर कर दीजिए तो आ जाएगा.
एक बार फिर से देखते हैं.
जी सर.
Is this entire screen visible now?
Yes, sir.
Yes, sir. Yes, sir. Yes, sir.
Yes, sir.
Okay. Okay, then I'll continue then.
Okay, sir.
Thank you. Thank you very much. Yeah. Yeah. So good afternoon to all of you, and thank you the organizers and Professor Yadav and others, his team, for for inviting me and for this talk. So the topic which I have chosen today is AI in healthcare. So here I'll be discussing some of my work that we have done in our lab. So I'll be focusing on that part only. So with the introduction, I hope this, since artificial intelligence in healthcare, so I hope this, what is artificial intelligence should be clear to everyone. So can anyone tell me that what they mean by artificial intelligence? So for me, it's the simulation of human intelligence, which means that to make the computers or the machine as intelligent as human. So this is the direction in which we are proceeding now, and this artificial intelligence can perform many tasks faster and more accurately. And this machine learning and deep learning are just the ways, or you can say techniques or tools to achieve artificial intelligence target. So one question I pose before you is, what is the limitation of human mind? And where we go for this AI? See, for all problems, we don't go for AI. Like suppose if you have to do sorting or some sort searching or this payroll calculations, we don't go for AI. We go for AI for some type of problems. So what are those type of problems where our mind has some limitations? If I ask that, suppose you have to recognize a person, can you explain the same thing, that how you have recognized a person? It's really a tough job to recognize, explain the other person that how you have recognized him. So this information is not as easy to pass to the other person. Similarly, naming of colors. So based on learning, not on absolute, that is not on absolute standards. So here in health, I'm going to focus not on my first work, that is not on human, but on animal. See, lots of work has been done on human health, but few work has been done to monitor the health of animals. So my work has been focused on animal health, where I've got very good funding for that also, for this work from ICR. And I'll tell you details about those things. So here this animal health monitoring refers to continuous observation, assessment, and management of an animal's health status to detect disease and prevent outbreaks and ensure overall well-being. And this involves tracking vital signals, behavior, nutrition, and environmental factors to maintain animal health, which improve productivity and reduce risk associated with infections or other medical conditions. So what is the importance of animal health monitoring? It prevents disease outbreaks and it enhances livestock productivity. Further, it reduces economic losses and ensures food safety and supports biodiversity and ecosystem balance. The key aspects of animal health monitoring are disease surveillance, where detecting and preventing infectious disease in livestock, pets, and wildlife, and behavioral monitoring, where observing the activity levels, eating patterns, and movement to identify health issues. Further, biometric tracking, where technologies like wearables and AI for real-time monitoring of health is required, and environmental monitoring, which analyzes factors such as temperature, humidity, and water quality for farm animals. And it also involves this telemedicine and remote diagnosis, where using smart web systems to diagnose and treat animals. So Here we'll talk about that how we have applied this deep learning for behavior and health monitoring of animals. Here we use computer vision to keep track of animals' behavior and monitoring, and we have installed a camera in farm to collect video data as well as the image data. And this eliminates the need to maintain physical devices on each animals. But before diagnosis any person or the animals, what is required? As you can see in the diagram, can some of you tell that what is this yellow tag there is in the ear of the cattle? See, these tags are there to identify the cattle. See, wherever the patient goes to the doctor, what is the important thing? See, each patient has unique number, and based on each individual, the medicine is given. So here also, unless and until we are able to identify or recognize the animal, then we cannot treat the animal. So whether we are treating the same animal or different animal, for that we have to recognize the animal. So our work is focused on this only, apart from other things. So what we have done, we have used animal biometrics to recognize the animals, each individual animal. And further, I'll tell you that how we have done these things, recognizing each animals. And I can tell you, we were the first one to start this task. And can you tell me that how these images are related to this? And for doing any work, we need some real life problems and motivations. So these are the good statistical data which shows that why we should go to recognize an animal and do the health monitoring of animals. So with this data, you can say that how much this contribution of this livestock is there in our GDP. And there are many cases of this missing and swapping and other things that can also be solved if we are able to correctly recognize the cattle or the animals. So these are some of the real life motivations for this, where this health monitoring and outbreak of critical disease are very important, which we are focusing here in my talk. So here this vaccination and deworming and treatment of animals and testing and outbreak of disease, these are important things which we are focusing. And these are some of the classical methods, or you can say traditional methods, that are used to recognize the animal. So before going to this, I'll just tell you that what are the different techniques that are there. One is the ear-based identification and tracking approach that I showed you. And as you can see that each animals are given some tags in their ear, which uniquely identifies them. So now for this AI, or you can say machine learning or deep learning, we need some data. Because if we have data, then only this AI or machine learning or deep learning works. So what type of data is required to recognize the animal? What do you think? What is your idea that what type of data may be required to recognize the animal? So here the selection of data to be acquired should be uniquely distinguish one animal from another animal, as in the case of human being, because we recognize the human being through faces or many other things, through fingerprints or face or iris, as you see in the Aadhaar card. So the same here also we have tried to uniquely identify each individual animal. For example, as you can see here, that each individual animal has some unique patterns or mark to which you can uniquely identify them. As you can see, this penguin has some patterns here on the stomach. This birds have some unique patterns at the beak. Zebra has some unique coat pattern here, as you can see here. So all animals are there. But our work has focused on cattle only. Initially cattle, then we moved to small animals. So as you can see here, this is the picture of muzzle print. Muzzle print is the mouth part of the cattle. And we were the first one to recognize that and capture the data and test it. And we have got the patent for this. And you can go just Google it and you can see our work related to this and the patents that have been granted to us for this work. As well as we have got very good funding from ICR, Indian Council of Agricultural Research, to continue this work. Now, this is one of the traditional methods, just it's taking through ink print, the muzzle print, and then trying to identify. But we have not done this. We have automatically captured the images through our mobile camera and tried to do it. So there are two things that is there in recognition. One is identification and the other is verification. Identification is who is this individual animal, or you can say from the given data sets, which animal is this one. You can say one-to-many matching. Whereas in the case of verification, here we test that is the same individual, or you can say one-to-one matching, like Aadhaar card we do. In Aadhaar card, what is happening? That our fingerprint is matched with our own fingerprint. It does not searches all the database and try to come out. So this is called verification. So in our work, we have tried to do both, that is identification as well as verification. So see, for database preparation, it is itself is a challenging task, but we have collected the database from this Institute of Agricultural Sciences, BHU, and we collected the muzzle print as well as the data sets of face images as well as what you call muzzle print for around 500. Not only additionally, we collected data sets from Indian Veterinary Research Institute, IEEE, Bareilly also. So totally we have now some 500 data sets. So as you can see that these data sets are categorized into different breeds of cattle. As you can see here, this baseline cow, this about 150, and hybrid Omlo cow also 50, and then Holstein Friesian cow. So these are all the data sets that we have collected, and for each one, we have collected 10 images each. So in that way, you can say the total 500,000 images were there to train our model. These are some of the sample images, as you can see here. On the left side, you can see there's a muzzle print. On the right side, you can see the face images of the cattle. So one more thing I would like to tell you, that this collection of data base is not the easy task. Why not easy? Because getting the front position and for the muzzle print as well as the face images in front position and proper light condition is not an easy task. So it took some time and effort to collect this data. So, but we collected this data and showed some very good results based on those. So this is the model that it is used for animal biometrics for animal that for any cattle or animals, or if for human face also the same technique is used. So this graph diagram demonstrates that. So this is the flow diagram of this cattle identification system. That is, sensor is there. Sensor means camera. Then some pre-processing is done, and then feature section, then template generation, then score matching is done with the stored biometric data, and finally the decision is taken that it is identified or not identified. This is how our machine learning model or algorithm works. These are few sample images of the cattle you can see, which we have used to train our model. So here we have used this computer vision framework for recognition and validation of cattle using face images, and you can see that here we have done this what called pre-processing, then feature section, and then uniquely generating the. So this is one of the work that where we have used this Gaussian framework, that is Gaussian level at different levels. So this is the work that has already been published, and here continuation of the same thing, that here it demonstrates that how the data is acquired, and then we have used this. These are the old techniques now, because LVP and this what you call HOG and all the old methods, but we still initially we started with that and we got very encouraging results. This is the sample images of the what you call muzzle print, for which also we. So here also I can tell you that we were the first one to recognize that the muzzle print is somewhat similar to our fingerprint. So we tried to figure it out and applied the algorithms that were used to recognize the fingerprint patterns, and we tried to customize it. So we were somewhat successful. Apart from that, we used some other techniques also to match different muzzle prints as we do in case of fingerprints. Here, some of the techniques that we used here, that it saved, served, all we tried and tried to match the inertia points of the inertia points, that is x, y, and theta coordinate values of each one. So these are some of the sample images. This is the upper part, middle part, and the lower part. So these are some of the intermediate results that is available. So this is the input muzzle point images, and as I told that we used number of feature section techniques like early texture features, color features, and hog feature, LVP, shifts, many we used and tried to get the good results, and then tried to recognize the identity of each individual animal. Here also this diagram shows the original muzzle point image pattern as well as segmented muzzle images with Kami's color-based segmentation approach. And these figures shows the segmentation of muzzle point images as well as the original images and brown tooth, and illustrates the finding of the. These are also some examples of muzzle patterns. You can say this left side is the calf muzzle point registration, and which is not very clear because it's not very much developed. And right side you can say muzzle point registration. So you can say that muzzle patterns of cattle are uneven features of their skin surface. They are different for each other, as like fingerprints of our human, as I told you earlier. And muzzle print or nose print is used as a recognized pattern for animals identification. For this reason, they can be used to identify cattle. As you can see here, we have used this single shift key point selection for muzzle print images, and we have used this 16x16 gradients and 4x4 cells for each. Here you can see the muzzle print, where the muzzle patterns processing are left from left. You can see the original image, reprocessing, and then overlapping display. These are the three images. And the approaches used for cattle identification are this local binary pattern, and as you can see here, this local binary pattern operators assigns labels to every pixel of image by thresholding the 3x3 neighborhood of each pixel with center pixel value and considering the result of number. We also applied this convolutional neural network for cattle. This, as you can see here, this is an example that we know the CNN models, how it works. Here we don't require from feature section to classification, all are done in one go using this. Similarly, this is for muzzle print also. As you can see, the left side, it is verified and this is not verified. This is how we get the results. So these are some samples just showing that if the muzzle prints are dirty or something, then this is also a challenge for us. And if the muzzle prints are dirty, then it's sometimes what happens that it may not recognize correctly. So before taking the images, we have to clean the mouth part or the muzzle of the cattles. So this is a close view of the muzzles. As you can see clearly, this beads and ridges are available for each cattles. So this is the proposed cattle identification method where we have used muzzle print and then different models. So I'm showing that because we published some of the papers. So I've taken these images from my papers only, and I'm showing it to you. So here also it is one of the work where we have used face images for different works. And this is how we have done. So here you can say that the project that we have got is about to construct a lightweight deep learning model that can be deployed on edge devices such as mobile phones and drones, which can verify two images and belong to the same cattle or not. So this is the project on which we are still working. And this project is not only for cattle. They asked that since we have already done some work on the cattle, they asked us to do it on the small animals. Small animals means they asked to recognize the sheep, goat, and pig. So this is the task that has been given us by the Indian Council of Agriculture Research. That work is still going on because the data collection is done by Indian Veterinary Institute, Bareilly, and we are doing the model training part. So this is some of the state-of-the-art techniques that we are still doing, where we are using this Siamese triplet model, and to recognize where we are Siamese triplet model, we are at the same time we are taking the muzzle as well as the face both and trying to recognize the animal. And we have got a very good accuracy. Now the problem challenge is that we have to deploy it also. These are some of the training results. Similarly, this is also one of the work. This is a proposed model for this pairwise triplet cross. So you can say that working on novel approach for cattle verification using deep learning methods, and here data efficient vision transformer model is being considered for future work that we are still working on. And we are also working on the XAI, that is the explainability part, so that our results are justified and explain that why we are getting these results. This is one of the proposed AI models that we have used and had been published in the some reputed journals. So I'll just give you this collaborative project about just 2 minutes talk about our project that we are still doing along with the Tanuvas Chennai. So there are two. The amount of funding here is around 2.25 crores, where 1.10 crores we have got, and some similarly this Chennai have got half part of money, and we are doing. And objective is that they are doing the clinical part, and we are focusing on the precision livestock farming and some health related issues. And they are doing some clinical, working on some clinical parameters of the cattle. So this is how our objectives have been defined. Here the first objective is to develop smart EVMR system, that is, you can say electronic veterinary medical record system, and IoT tools and wearable sensors for medical data capture and monitoring applications in animals. And here we are also looking for AI tools for veterinary syndromic surveillance and disease monitoring. And fourth one is smart farming and healthcare apps for ruminants and pet animals and smart telemedicines. So these are the work that is still going. 1.5 years has passed, 1.5 years still left, and we are on the mid of the project. It is still being done. So here what we are doing is that we have doing this, making smart electronic health records for animals, for animal owners, as well as electronic veterinary medical records system for vets. Here, some prototypes of electronic health records for animals and electronic veterinary medical records are already developed, and health and medical data architecture module and data panels are also developed. So these things we have already done it collaboratively, means jointly we have done with Tanvas Chennai and at IIT BHU and this IVRI Bareilly. So they are also using some of the IoT devices also to capture the and some wearable devices for the animals, and they are still collecting some data sets, and they're also working on some improve the productivity of them, what you call the meat production. And as you can see here, that it is a goat, and so we are doing, as I told you, that we have got this for goat, sheep, and pig, both, all the three. So here you can see the trace of using AI to predict body measurement and its utilization for body weight and calculation of sheep and goats. So these are all things are done. And the data is still being captured and the model is still being trained. So the objective 2 of this project is to develop artificial intelligence-based smart system for precision livestock farming, where that involves here this creating a data sets based on muzzle imprint and facial images of individual animals to develop unique digital signatures for sheep, goat, and pigs, and developing a smartphone-based app for recognition of each animal using facial images, as I told you earlier. And the other activities involved is we are developing some deep learning models, which involves digital signatures and testing performance of biometric authentication systems and generating unique ID for each farm animal. And we are also using thermal imaging-based recognition biometric authentication system. And apart from this, we are also using smartphone-based recording and retrieval production performance data for animals for livestock farming. As I told you that we are using this face and muzzle print images for this. And this objective 2, we are also doing this, that some challenges are also there, that the collected images are processed and sometimes there are some noise, occlusion, and intra-class variations. So these are some of the challenges we are also facing and trying to overcome those things. And the images that are analyzed through a hybrid model of residual attention network and multi-scale contextual. These are some of the models that we are using for to recognize the cattle. And model is developed based on muzzle prints. We also utilize for other animals. And we are also developing thermal images as imaging-based recognition, and that work is still going on. So here you can see that some of the data recording that we are doing at, for each individual, you can say cattle or the small animals. As you can see here, this is categorized into general information, growth parameters, production parameters, reproduction parameters, and farm input records. So here you can see in general information, we are collecting this animal number, species, date of birth, sex, then SIR number, dam number, exotic inheritance. So these all data are being collected under general information, and both parameters that includes body weight, birth weight, weight at first heat, and weight at first conception, weight at first, and etc. Similarly, for production parameters, we are using that age at heat, AI per conception, service period, and calving interval. So these are all the some of the production parameters. Similarly, this reproduction parameters are also there, and farm records are there, and vaccination status and deworming status. So these are all data are captured or acquired. So here you can see this is how the images are taken. frontal view, side view, and other views. So through which we are able to, this you can say that at least 10 images are required, which involves front view, full left view, full right view, left facial view, right facial view, and muzzle print. So these are all data is collected for each individual cattle. Similarly for goat also, or for other animals. This is how, this is the flow chart, that how it is being done. We are also using this eyes data to record some of the health parameters, because this is also the first indicator of some issues are there. Then through eyes also, this can detect some diseases. So this is also being done. Apart from recognizing the each individual this sheep and goat. Similarly for pig also, we are doing this, capturing the data and doing the same thing for pig. So this is overall the project output, where we are using muzzle print, face, and we have promised that smartphone-based apps for real-time monitoring and retrieval of farms records, and smartphone-based apps for providing reminders and yeah. Can you hear me, all of you?
Yes, sir, you are audible.
Achha, I'm audible. So okay. So my speed is okay, or should I go slow or fast, something like that?
Sir,
Okay.
sir, it's perfect. Very perfect.
Okay, fine. So these are some of the publications of this work. So yes, we have patent has also been granted, and good number of publications have already been done for this area. Now I'll move to my next work related to health only, that involves bioinformatics as well as, you can say, drug discovery part. So as you know that biological data, for bioinformatics data, biological data involves DNA, RNA, amino acids, and proteins. But we have worked on at the level of amino acid chain, and I'll show you how we have done those things. So, yes, so you can see here, this is a biological database. This is a sequence database and structure database. Both are here. And in sequence database, you can see this nucleotide sequence and protein sequence. So we have worked on sequence only, data only. And the database that we have used is the open data set. And we have tried to solve an important problem. You can see that since all of you know that there's a problem of what you call antibiotic resistance. So we have worked on those problems. Here, this what you call this diagram shows the concept of genetic code and amino acids and proteins and peptides. See, for us, they're all textual data, or you can say the NLP. So whatever the models that are applied on this NLP or the text, yeah, that can also be applied here also, because these are all text data. So we have basically focused on the proteins and peptides. So why these peptides? As I told you, that this peptide bond is a covalent bond, and that is formed between two amino acids. And the advantage of this peptide is that it is extracted from animals and plants. I'll give you a good example for this. That have you seen that any animal taking medicine, how these animals treat themselves? See, even if you take a frog out of this water and cut and put it again into the water, what will happen? After some time, you will feel that it heals and then becomes all right. How these things are done? See, this because their body contains peptide, and that is the similarly to other animals also and plants also. They have the peptides inside their body, and through that they. So this is the new area that is being explored, that and which is being considered to be the alternative of the antibiotics, because antibiotics have certain, we can say, disadvantages. It has some side effects, as well as our body is getting resistant to that. I told you this. So you can say that antimicrobial resistance now causes more deaths than HIV and AIDS and malaria. So here this work, our work was to predict some molecules, and it was tested at the IBRI and at the laboratory level. And you can see that, some 20 amino acids are already available, and you can say, and it's still being used for internal use, but for wound healing and other things, and some creams are already available from peptides. So here the problem of antimicrobial resistance is that there are many sources or causes. One is the natural cause, that is mutation or gene transfer from one species to other, and some man-made causes are also there, because sometimes inappropriate use of antibiotics, and sometimes we take antibiotics and live in between. That also creates antibiotic resistance. So there are many reasons to develop this resistance towards antibiotic resistance in our body. Here through this diagram, you can see that, as you can see, that how this antibiotic resistance develops. You can see that from animals get antibiotics and develop resistance bacteria in their system, and then person gets antibiotics and develops resistance bacteria in his system, and similarly through food and other plants that we consume, that also sometimes tries to helps in increasing the antibiotic resistance. So what is the solution? The solution is peptide-based antimicrobials. This is what we have done. We have tried to predict some of the antimicrobial peptides that can be used, and for different ones, that is for antiviral, antibacterial, what we call cancer. So all peptides are there, some combinations are there, and we did work on some of them. So as you can see here, that's the advantages are that broad spectrum of antimicrobial activity and effective on traditional antibiotic resistance strains, no or less toxic side effects on humans, and do not induce drug resistance easily as compared to the conventional antibiotics. So the question is how they can help. So being a researcher, we can develop computational tools for this, and this could help save time and effort of scientists who are trying to synthesize them. So these are some of the antimicrobial peptides and the classifications. As you can see here, the sources are, this is amino acid rich species, and you can see the some structures and activities of these antimicrobial peptides. So availability of MPs from different sources are, you can see the maximum is from animals, then plants, then from, you can say, bacteria, then others, very few from this fungi and archaea. So as you know, this when you go for some drug discovery, you have to predict some molecules that has to be generated into from in silico to in vivo and then in vitro. In vitro means testing on some animals or some other things. So this is how it is done. So how to apply deep learning algorithms in this field? So what we have done is, we have every peptide is represented as an alphabetical string by a chain of standard amino acids, and there are 20 standard amino acids which are represented by letters of the English alphabet. So you can say that these are just like text for us, or it is an analytical problem for us. And here these algorithms like SVM use quantifiable properties of peptides such as physiochemical structure and constitutional features as predictors in order to classify them as antifungal or otherwise. And on the other hand, raw peptide sequence can be directly modified using conventional deep learning architecture like recurrent neural networks, convolutional neural networks, to build the robust classifier. And here you can see that the deep learning classification and discovery tools for AMPs, it involves the same process, that data collection, data pre-processing, and then construct embeddings. Embeddings are required because what you call, each text has to be given some unique what you call, some numbers, because computers cannot process this textual data, so it has to be converted into some numerical values, and then find feature matrix for that. So finally, this train and tune the model and test the module's performance and discover AMPs in proteins. Like this, take a random protein sequence and then from a set of possible peptides that can be extracted from it, and these peptides model and get prediction scores for each of them. And peptides with higher scores, let's say 0.5, can be considered to process antimicrobial potential, as seen here. Greater than 0.5 and less than 0.5. It's non-AMP antimicrobial peptide, and is greater than 5 is antimicrobial peptide. This is how it is done. So we have already developed some web-based applications where any researcher can go and log in and test that whether the peptides are antimicrobial peptide or antifungal peptide. So these things are already there, because for publishing papers we have already built some, and these are some of the publications that we did related to this work of peptides. See, these all works are done by my research scholars in my lab, and then the final testing is done at the Indian Veterinary Institute, IEEE Bareilly, because we have a very good collaboration with them. Now I'll focus one of my more work that is also the data here we got from IEEE, that is the canine data, that is dogs tumor data, whether it is cancer or not cancer. So here this, before starting, that has shown some of the successful stories of this, what you call, medical images that already has covered. So you can see that this wide applications of this deep learning models in medical images. So you can see that it has helped in real-time clinical diagnosis, a whole body portable ultrasound, radiology assistant. So these are things already available or developed. And for skin cancer also, lots of work has been done. And these are some of the success stories of medical images. So now the question is that what we have done. So I'll come to that point. That so medical imaging, you can see that medical imaging is defined as non-invasive visualization of internal organs and tissues. And medical image processing analysis, you can say, if you take the example of radiology or X-ray, ultrasound, or CT images and magnetic resources, find that this MRI or cytology domains have been major research fields for several decades and still continue. And these factors make it rather difficult to extract regions of interest, that is nuclei and cytoplasm, with segmentation approaches. And the work that we have done here is related to the histopathological images of canine, and that we have compared with the human also. So the motivation for this work is that you can say that cancer is one of the big challenge, or you can say major problem, and deaths are happening because of these cancers. And canine memory tumors, that is in dogs, are excellent models for human breast cancer and have high incidence and mortality rates. And you can say that diagnosis of both human breast cancer and canine memory tumors is made by histopathological images diagnosis, and and that are at an instant tissue sections by skill pathologists, which is very tedious and time consuming. So if we are able to develop some model and good things, that will help increase the efficiency of the doctors. So as in humans, because of due to increased awareness about disease, early diagnosis is somewhat possible with the help of routine cell checkups, mammography, followed by biopsy. So this is how we have done. So the left, you can see this gross and laboratory examination of CMT and surgical removed by biopsy. Then the tissue is removed, and then tissue embedding in paraffin block, then tissue sectioning, then slide staining. Then it is sent to this microscope, and finally this image. So this is the whole process that demonstrates our image acquisition. These all tasks has been done at IVRI, and our work started after getting the images. And you can see the different types of what you call carcinoma. That is cancerous images, carcinoma is cancerous images, histological cancer images. And this diagram shows one of the methods that we have applied, that is VG60. Train the model and for extend the features, that is the pre-trained model. Then we applied some classifiers, and then finally classified into benign or malignant. So this work was also published in very good reputed journal. You can see, go and see my this what you call my through my web link or my Google Scholar, you find all these papers there. And these are some of the encouraging results of that work. As you can see here, we have used this VGNet 16, and along with that, we use some different classifiers. We did not use full VG16. We used VG16 only just to extract some features, and after that, some classifier was used, mainly this osmium with different combinations. As you can see, RBF, polyene, and lead, and results are really encouraging. This is the AUC curve. So the problems that we addressed during our research was to design and develop an automatic system for recognition of cancer, and we validated the prepared database of CMTs. That is, you can say, tumors for recognition and verification of cancer on existing computer vision and deep learning approaches, and investigated the effect of data augmentation, stain normalization, and magnification on the performance of proposed work. And the proposed framework resulted in mean accuracies of 97 and 93% for binary classification of human breast cancer and CMT respectively. As you can see that CMT's and scanner immunity accuracy is somewhat less than the human. And the problems that we addressed during this is we designed and developed automatic system for recognition of cancer, and validated the prepared database of CMTs, and investigated the effect of data augmentation, stain normalization, and magnification on the performance of both framework. And these are some of the publications that we did related to this work. So now I'm going to conclude. So if questions are there related to my talk, so people can ask me. So what I suggest that if you want to start your journey, so you can, if anyone want to start work this AI machine learning, so he can just start by going through these stages. If they is just starting, intermediate, advanced, or mature, so they can follow these things. This is just suggestion. Anyway, these things you can find anywhere. So this is just my suggestions. And these are some of the resources that is available. So I invite questions from the audience or the business scholars or the students who are listening this my talk. Any questions?
If anyone have any doubts, he or she can ask directly.
Yeah. These are some of the facilities that are
In
available.
chat box. Any one of you? So there is one query from Rohit Agrawal, sir. Can we get such PPTs? So I will request with the experts after the completion of this FDP, and definitely we will share the relative PPTs with the.
Yeah, I'll share my PPT. No issue.
Any question? Neha,
So the students are research scholars or some BTech students or?
sir, these are the different faculty. These are faculty members from different institutes, including few research scholars.
Okay. Yes. Okay.
May I conclude it?
Thank you so much, sir, for your time, knowledge, and insightful presentation. It provides a fresh perspective on emerging areas in computer science. With this, we conclude second session of day 1 of our faculty development program. Thank you all the participants for your active engagement and patient throughput. Thank you. Thank you, sir.
Thank you. Thank you to all for listening me. Thank you.
Thank you very much, sir.
Thank you.
You have accepted our request, and from your busy schedule, you have give your valuable time, and definitely it will very fruitful for all the faculty members who
Yeah.
are joined here. Thank you so much, sir, and definitely in future, we hope that we will get your
Yes.
blessings again. Thank you so much,
Thank
sir.
you. Yeah. Thank you. Thank you. Thank you to all of you. Bye.
Sir, just leave.